Let's take a look at the science of life. The science of biology is the one that combines chemistry and physics and applies them to living things. A great place to start looking at biology is to start with a seed. We've all seen seeds. We have all planted seeds. We have certainly seen what happens when a seed grows into an organism. A seed is really a container for a plant embryo, and that plant embryo is made of cells just like plants are. And those plant cells are not that much different from other plant cells. They have a cell wall surrounding them, a nucleus surrounded by both smooth and rough endoplasmic reticulum in the center. Within the nucleus, there's a structure called the nucleolus. There are mitochondria, so that you can make ATP using glucose as a fuel. There is um, Golgi outside the endoplasmic reticulum with its associated vesicles. There are ribosomes, both free in the cytoplasm and on the endoplasmic reticulum, the rough. And there are vacuoles. And if we took a closer look at the cell membrane, not the wall, the membrane, we would find the phospholipid bilayer with the lipid tails pointed together and the phosphate groups pointed outward. It would also have cholesterol molecules embedded in it, transport proteins moving things in and out of the cell membrane selectively, and carbohydrates attached to it. Plants grow from seeds by the cell cycle, the mitotic cell division. So we go from one cell in a seed to two cells in the seed, and eventually those two cells will grow into the entire plant organism, a marigold in this case. That is the mitotic cell cycle that starts with interphase and then goes to mitosis and cytokinesis. In interphase, we have three parts: G1, normal growth; S, when the DNA is copied; G2 for last minute growth; and then mitosis, which goes prophase, metaphase, anaphase, and telophase, separating the two nuclei. And cytokinesis splits the cells into two new cells. So, where does the energy come from to power life in the cell, and for that matter, in the organism? We know it comes from the sun, specifically plants using the chloroplast organelle. Sunlight hits a chlorophyll molecule and causes it to lose an electron. That electron goes to an electron transport system, which, like all electron transport systems, makes ATP. But this one only makes ATP for the chloroplast. As it is missing an electron, chlorophyll steals one from a water molecule, causing hydrolysis, in which the hydrogen ions go into the electron transport chain, and the oxygen finds another oxygen to make oxygen gas. In the next step, a molecule called Rubisco combines with a carbon dioxide from the atmosphere and breaks down into two 3-carbon molecules called PGAs. The PGAs then use the ATP from the light reactions to be rearranged into two simple sugars called PGALs. Most of the time, 5 out of 6, those PGALs are converted back to Rubisco. This process is called the Calvin cycle. However, 1 out of 6 times, the two PGAs join and form a glucose molecule. This glucose goes to the cytoplasm, where it will fuel the other energy converting organelle, the mitochondria. The glucose in the cytoplasm first undergoes glycolysis, the splitting of glucose into two pyruvic acid molecules. Each of these pyruvic molecules will then lose a carbon dioxide to become an acetyl group that will be picked up by coenzyme A. In the mitochondria, the acetyl-CoA will pass the acetic group to a molecule called oxaloacetic acid and create citric acid. Citric acid will lose a carbon dioxide to become ketoglutaric acid, which in turn will lose a carbon dioxide to become a succinic acid. Succinic acid is then rearranged into the starting oxaloacetic acid. This process is known as the Krebs cycle. So far, not much ATP has been made, but what we do have are charged coenzymes. These carry high energy electrons from the Krebs cycle to an electron transport system. In this system, the electrons are passed from molecule to molecule, and with each pass used to make an ATP. The last thing to accept that electron is an oxygen atom, the same kind of atom that lost it in hydrolysis. This electron transport system can produce about 32 ATPs from a single glucose molecule, and it is this ATP that is used to power the everyday activities of the life of the plant. One of the everyday jobs that cells do is to build the proteins that do the work in the cell and build the organism. This starts with the process of transcription. In transcription, DNA helicase opens up the double helix at the gene. Then RNA polymerase builds a messenger RNA transcript which contains both introns and exons. The introns are removed, the exons are spliced together, a cap is added, and a poly A tail is added to the other end. This mature messenger RNA is then exported out of the nucleus into the cytoplasm. Once in the cytoplasm, the messenger RNA finds a ribosome and begins the process of translation. Transfer RNAs bring in the correct amino acids and peptide bonds form between them, creating a growing peptide that will become the mature protein. That protein will eventually be released, moved to the Golgi body, and delivered in a vesicle to where it can do its job. When a marigold or any organism reaches a certain age, it prepares to produce offspring. In plants, this occurs in the flowers. There are both male and female parts to a flower, and each produces gametes or sex cells. In the case of plants, these are called pollen and egg. The process is similar in each case and is referred to as a meiotic division. It's a reductive division because the cells created are haploid. Haploid means they only contain one copy of each chromosome instead of the normal two. These cells are then able to combine with an opposite sex gamete to produce a stem cell that will grow into the new organism. The steps of a meiotic division are very similar to mitosis, but in meiosis we have new structures called a tetrad composed of 4 copies of each chromosome. One copy of each in the end will be distributed to one of the 4 gametes or sex cells produced. In this diagram, the chromosome is represented by the gene T. So at the end of meiosis 1, there are 2 copies of T in each cell, and at the end of meiosis 2, there is only 1 copy of T in each gamete. Unlike most animals, but like many plants, marigolds can be self-fertilized. Knowing the genotype of the marigolds makes it possible to predict what the offspring of the plant will be. This is really thanks to the work of Gregor Mendel, who gave us the law of independent assortment and the law of segregation. Reginald Punnett also made our lives easier by devising Punnett squares. If we examine the T gene, we know that in both cases, pollen and egg, half of the gametes will contain a big T and half will contain a small t. One gamete goes across the top, one down the side, and then we fill in the boxes. From this, we see that we get a homozygous tall, 2 heterozygous talls, 1 homozygous short, for a ratio of 3 talls to 1 short. Therefore, if you plant seeds from this marigold, that's what you would expect to have grow. This may not make a lot of sense to you today, but over the course of the next 9 months, we are going to become very familiar with each process outlined. There is a lot of work ahead, but in the end, you'll have learned a ton.